Namaste to everyone. I'm Tara Pokhriyal. I'm from India. I have completed my MSc or Masters in Biochemistry. Here I'm presenting my research work which I have done in Masters, that is effect of heavy metal ion on the antioxidant properties of Mentha spicata. A bit introduction I want to give to all of you, that in the recent year we all are aware that antioxidants are increasing day by day. Why is it? We all are aware about antioxidant. Antioxidant, the reason for going behind antioxidant is nothing but it can protect our cell, their organelles and other metabolic pathways which are occurring in our body, or we can say the biological system. They destroy all the reactive oxygen species which are generated in the biological system. These ROS we all are aware that produce in a biological system, but how? A physical and a chemical toxin. They are responsible for the production of reactive oxygen species. When they entered in our biological system, they are produced, and the overload of this ROS increases the concentration of there, because of which several diseases occur, which is they cause damage to the DNA, lipid, protein, chromosome break, alteration in signal transduction, and gene expression occur. And because of which they implicated of various disaster diseases, we can say that the cancer, the cataract, birth defect, reproductive anomalies, and various neurodegenerative diseases. So there should be a balance between the production and the removal of this ROS, is it? Biological system. When we are talking about biological system, our biological system is endowed with an array of antioxidants, but these antioxidants are not adequate for the damage, for the neutralizing these ROS which are generated in our biological system. So there is always a need of an external source which has given them antioxidant. And nowadays fruits, vegetables, spices and various other herbal resources, they are responsible for the external sources of antioxidants. Now we are talking about antioxidants. What are these? These are nothing. These are the compounds which are used to detoxify or neutralize the free oxygen species which generated in a biological system, nothing else. Here, in my research work, keeping in mind the antioxidant potential of mint and the role of abiotic stress in activating plant defense mechanism, I have studied the effect of heavy metal ion on antioxidant potential of Mentha spicata. Now, we all are aware that plants are immovable. They cannot move from one place to another. Is it true? Yeah, it is true. But so they are subjected to a various abiotic stress such as drought, temperature, salinity, soil pH and various other, and most effectively is heavy metal ion. This metal toxicity can cause a redox imbalance in a biological system, and due to which various neurodegenerative disorder can occur in our body, which is a very serious condition, I must say that. Now, for that, plant have developed various strategies to cope up with these type of imbalance occurring in their biological system. Some plants use avoidance strategies and others use the internal defense mechanism, that is nothing but antioxidants. So, now we can give a definition of antioxidant. Antioxidant detoxify, neutralize, or metabolize reactive species and hence reduce the incidence or severity of degenerative conditions. Okay, what is the objective of my study? It is nothing. It is to determine the change in the total polyphenolic compound, or we can say the content, TFC, that is total flavonoid content, and to characterize the free radical scavenging effect on ferric ion reducing capability of herbal plant, that is Mentha spicata, which have given the heavy metal stress. Secondly, we have done the comparison of antioxidant activity which have given the heavy metal stress. Thirdly, we have validated the result, and then we have come to a conclusion and have given some results. Now, what is the experimental design of my research work? It is, first we have done the plantation. Next, we have done The heavy metal treatment. We have given heavy metal treatment to the plant which were taken as a experimental plant. Thirdly, we have done the sampling. Next, we have done the extraction. Then we perform various assaying method, and after that we come to a conclusion. We analyze our finding and come to a conclusion and give us a result, which is very surprising. The first is plantation. The plant variety which we have taken is nothing but Mentha spicata. The number of units which was taken is 30 units. And these 30 units were taken from the Punjab Agriculture University, Ludhiana, India. Now the next question arises is why Mentha spicata? The answer is very easy. Mentha spicata has a short life cycle. It is a perennial herb, easy available and has a low maintenance. And it is also having a high phenolic compounds which is responsible for the antioxidant properties of Mentha spicata. Because these phenolic compounds have hydroxyl group, this hydroxyl group is used as an hydrogen donor to a plant. What we have done? We have taken the plant and raised to them in a poly bags which is containing a 1 kg of garden soil. Then we have divided the plant into 6 groups, each group having 5 plants. 2 groups of plants were kept as in control, that is they were given a normal plant watering. The other 4 group of plants were treated with the heavy metal. Now the heavy metal treatment, what was that? Different heavy metal at the rate of 10 ppm were given. Lead as lead acetate, cadmium as cadmium sulfate, arsenic as sodium arsenate, and nickel as nickel sulfate. This treatment was not repeated. It was given only once. 500 ml of solution was prepared of each heavy metal, which is given 100 ml to each plant group. Then the leaves were then plucked after every 15 days of time for experimental purpose. Now sampling. We have done sampling after 15 days and after 30 days. That is first generation leaves and the second generation leaves. Now the extraction. We have extract the methanolic extract of this plant by taking 5 gram Mentha spicata leaves were grinded in a pestle mortar and it is mixed with 50 ml of methanol. Then we have allowed 1 hour incubation at room temperature. Then we have centrifuged at 4000 rpm for 15 minutes. Mixture then filtered by a muslin cloth. That filtrate is kept for an experimental purpose. Now the assaying method which we have performed for this research work. First one, we have done standardization. We have taken the standard compound for evaluating the result. Then we have performed the total phenolic content, then TFC, then total reducing assay, FRAP method, DPPH scavenging activity, and superoxide scavenging activity. What is standardization? We have taken gallic acid as a standard compound for evaluating phenolic content, quercetin as a standard flavonoid compound for TFC evaluation, and ascorbic acid for the antioxidant potential of plant. Now the method, these are the method. We have taken the 10 microliter sample, then we have added 490 microliter of water, 1 hour incubation, 500 microliter of Folin reagent, then we have added 2 ml Na2CO3, and then we have taken the absorbance. This absorbance is taken, is increasing. In TFC, we have taken the 100 microliter of sample, and we have added 500 microliter of distilled water, then methanolic, aluminium chloride, then 2 ml K2CO3, then 2 ml distilled water, and then we have taken the absorbance. Total reducing assay, 100 microliter sample, 500 microliter phosphate buffer, and 500 microliter phosphate. Incubation then we have given at 50 degrees Celsius, 20 minutes, 500 microliter trichloroacetic acid was given. 10-minute incubation, centrifuge, 2500 rpm, supernatant was taken, then we have added the distilled water to it. Then solution is made to 5 ml and we have taken the absorbance. FREP assay, firstly we have prepared the FREP solution, which is prepared by 2.5 ml of acetate buffer, 2.5 ml of TPTZ, plus 2.5 ml of FeCl3 6H2O, then we got the FREP solution. To that FREP solution, we have added 100 microliter of sample. Then solution is made to 5 ml by adding distilled water. Then we have taken the absorbance of that. Scavenging capacity, DPPH, 10 microliter of sample, that is the extract which we have prepared in methanol. Then 2.5 ml of tris HCl and 1 ml DPPH is added. After 30 minutes of incubation, we have taken the absorbance of that particular sample. For superoxide scavenging activity, we have taken the 100 microliter of NBT, NADH, and then we have added the 100 microliter of sample, then PMS is added, then absorbance is taken. Now the calculation. The sample values in TPC, total phenolic content, total flavonoid content, total reducing assay, FREP method are calculated in mg per grams, which is gram equivalent to the standard compound which we have taken for the standardization. The equation which we got from for the standard compound, that is from y is equal to mx plus c, where y is the absorbance of a sample, m is the absorbance of the standard compound, then x is the value to be determined, and the c is the constant value. This y is equal to mx plus c is different for every method, and accordingly we have applied to that sample and we got the result. For calculation of DPPH and superoxide scavenging activity, we have applied the formula, that is the value of blank and we have minus the value of sample, then divided by blank into 100. This is the percent inhibition we have taken. Now the result and analysis which we have done. Various assaying methods were performed by using ascorbic acid as a standard. TPC, TFC were measured as a gallic acid and quercetin equivalent. Total reducing assay, FREP, were measured as an ascorbic acid equivalent. DPPH and superoxide scavenging assay were examined by calculating percent inhibition and also examined by seeing percent change of the plant over the control. We have also taken the percent change over the control plant which we kept for the sampling. These are the results which we have got from our experimental work. The values in the parenthesis is showing nothing but the change over the control. The control which is showing that first generation leaves and second generation leaf. And the 28.5, this is nothing but it is the value in mg per grams. On the basis of that, we have prepared a graph, calibration graph, we can say that. For TPC and TFC, first generation and second generation control, we have seen that there is an increasing effect antioxidant activities. In total phenolic content, we see that as plant are adapt to the stress, so they are synthesizing more phenolic compound. So they are synthesizing more phenolic compound, so the activities increase, because of which the first generation is having high content and the stress. This is basically showing the stress. The first generation is having the high stress problem, but in second generation it was reduced because more phenolic compounds are producing in that particular generation. This is the total phenolic content, the percent change over the control which we have studied. This is for the total flavonoid content. This is also the percent change over the control which we have taken. Now the values in parentheses represent the change over the control, as you all know that. And the 47.5 which is showing over there in black form, this is nothing but mg per grams value which is taken as ascorbic acid gram equivalent. Total reducing assay, same thing is happening here. The reducing capacity is measured in total reducing assay and the FRAP assay. Here is the capacity we have measured, the reducing the power of ferric ion. In total reducing assay, we have taken the potassium ferrocyanide. We have taken the potassium ferrocyanide, and this potassium ferrocyanide forms a complex with the test sample, and because of that phenolic compound, the activities increases, and because of that increasing activity, the ferric ion is converted into ferrous compound. The complex which was formed by potassium ferrocyanide and the test sample is converted into ferrous compound. That means ferric from +3 state goes to +2 state. Same thing happens in FREP assay. Here also the ferric ion is converted into ferrous compound. Then the reading is taken. This is the percent change over the control which we have taken. This is for the FREP assay we have taken. Now the DPPH and the superoxide scavenging activity. The same thing, value in parenthesis, it is showing the change over the control, and these are the values which we have got from the regression curve of the standard compound which we have taken for our experimental work, that is ascorbic acid. The superoxide scavenging activity, as we all know that in the method slide I have shown that the PMS and the NADH is responsible for the why we have added the PMS and NADH. This is for nothing but for generating superoxide. This superoxide is generated, because of that the phenolic compound increases, and because of that hydrogen ion is donated to the superoxide ion and it get redox inactive, and the electron which is free from phenolic compound goes to NBT, and that NBT is reduced. The same thing is happening here and to every procedure. This is the graph showing the scavenging activity of DPPH and superoxide. The activity is basically increased in second generation. This is the finding which we have got. This is the % change over the control which we have got for superoxide scavenging activity. This is the % change we have got. Now the conclusion. Now we concluded that Mentha spicata plant test sample treated with different heavy metal ions like cadmium, nickel, lead and arsenic reveals that it contains antioxidant. The antioxidant activities of the plant samples are due to the presence of phenolic compound containing the hydroxyl group that confer the hydrogen donating ability. The antioxidant action of reductones are nothing but phenolic and the flavonoid compound which are present in the Mentha spicata plant. On the breaking of the free radical chain by donating hydrogen atom, reductant also react with certain precursors of peroxide, thus preventing peroxide formation. The strong correlation observed in the present study between antioxidant activity, phenolics and flavonoid content of different heavy metal on plant suggest a possible use of their parts in making the active ingredients of antioxidant supplement after removing the toxic ingredient if there are any. Thank you so much.
We still have time for questions.
I think I can continue. Thank you for the very stimulating presentation. In your conclusion, because one of the things I could observe from the presentation was that the samples spiked with heavy metals.
Sorry, I'm not getting.
That the samples that were spiked with heavy metals had
Heavy metals.
Yeah, had a higher concentration of antioxidants.
No, we have given the particular 10 ppm concentration
Yes,
solution to them.
Yes,
That
compared to the control.
Yeah, no. That was not for control. We have seen the activity of the plant has increased to heavy metals which we have given.
Yes, I know. That's what I'm saying. Yes, you have control without heavy metal.
Yes.
Yes, and the other ones you added 10 ppm.
Yes.
And the ones with 10 ppm had higher antioxidant.
No, we didn't studied about that. We have induced the 10 ppm concentration stress to the plant. That is all.
Yes, when you analyze this, the plant now, the leaves, after harvest, the antioxidant content of those plants were higher than control. Yeah, and the conclusion, your conclusion said that the plants made use of these heavy metals to produce the antioxidant. What are we? Do you think maybe the stress? Because antioxidant production, they are produced as a result of stress, the plant mechanism to overcome the stress. So a possible conclusion could be that the plants, in trying to protect themselves from the toxic effect of the heavy metals, now produce this antioxidant to overcome the effect of the stress, rather than making use of the heavy metals to produce the antioxidant.
But nowadays, because of industrialization, we are seeing that the heavy metal is disposed to any area. And because of that, plant are affecting. You know that.
Yeah.
And because of that, the heavy metal, plant has adopted an internal mechanism system. And
Yes.
one more thing I want to show that I have studied the non-enzymatic activity, not the enzymatic activity. The enzymatic activity is occurring in our biological system. So they are also the antioxidant, but we have focused on the non-enzymatic activity that was shown in this particular research. Is that clear?
So perhaps, do you notice that the lifetime of the plants got shortened by the effect of these heavy metals?
Mm-hmm.
Because you were saying that your plant was short life, right? So it doesn't last too long. So is there an effect on the life? I mean, how long it survives, or is?
No, that is why we have taken the plant which have a short life cycle so that we can perform our study. We didn't focus for the whether it is affecting its life cycle or not. We have only focused the heavy metal toxicity which we have induced to that particular plant.
No, I know, but after adding the heavy metal, so is the plant dying faster, or?
Definitely it will affect.
Okay.
Because the shedding of leaf may occur, the root accumulate the reactive oxygen species. Because of that, the plant can have the deficiency of oxygen, and because of that the plant may die.
Okay. So based on that, so according to because now that we know that the heavy metals are toxic because they are dying faster. So amongst all the heavy metals, the ones that you have used, you have lead and you have arsenic. You have had nickel and you have cadmium. So arsenic and lead are very toxic heavy metals, even more than nickel and cadmium. But your plots are showing that nickel has the strongest effect, and cadmium also has, in some of the charts, they have the strongest effect. So how can you explain, or how can we explain that kind of discrepancy, that even though that, the more toxic heavy metals have the weakest effect compared to the nickel and the cadmium metals?
Okay. First of all, we have not analyzed the strong effect and the weak effect of a particular heavy metal. We have only taken the different heavy metal for a testing purpose and nothing else. But this was a small scale project that was of 4th month, and in that particular time period we have completed only the effect of heavy metal ion on the antioxidant properties, not showing which is the strongest one and which is the weakest one having effect on plant. We have not studied that. Sorry?
That your slide that has a mathematical expression y is equal to mx plus c. I want you to tell me which one depends on the other. That is, the variables there, you have dependent variable and independent variable. Can you scroll down to that, the slide? Do you remember?
Yeah, I remember.
Y is equal to mx plus c.
Y is equal to mx plus c, basically nothing, but we have the standardization which we have done in our project work. That standardization work is done through that particular equation, and on the basis of that equation in the Excel we have prepared a chart, calibration curve we have prepared, and we got the finding y is equals to 0.017 x plus c, and the c is the constant value which is given inbuilt.
I know. I just want to ask, between the y and the x, which one is causing the effect, the causing effect on the other? Is it the one that is represented by y, or the one you represented by x?
I'm not getting your question.
Oh.
Because of your accent, I'm not getting it.
Sorry, maybe if you scroll down to that equation. Sorry.
Okay.
I think that's one. Okay. You said y is the absorbance of sample, and x is the value to be determined. Is it that where y is the one depending on x, or x depending on y? That's just what I want to know.
Because you are asking that
which one causes effect or changes in the other?
The X is the standard compound,
okay,
which has not given any stress. But the Y is the sample to which we have given the heavy metal stress. So we are taking the standard compound and we are comparing the antioxidant activities from the standard compound and from the sample which we have taken. That is why Y is equivalent to mx, because m is the value which is given in the Excel sheet because of that particular absorbance of standard compound. That is why we have equal to that particular equation.
So in that case, it is X that is changing due to Y.
No.
Okay. We can continue after. Sorry, we must finish our work. Thank you very much, Tara, for
Thank
your
you so much.
presentation. And I think it is discussion confirmed with.
Thank you.
Thank you very much.